Hi everyone, this is Ratna. Welcome to Basics of Biotechnology. In this tutorial, I am going to discuss about DNA replication. DNA replication. What exactly DNA replication is? It is a biological process of producing two identical replicas of DNA from one original DNA molecule. See this picture. DNA, this is the original DNA. It is synthesizing a copy of its own. It is nothing but DNA replication. In other words, we can say like it is a duplication process of DNA. It is duplicating itself. It is synthesizing a copy of its own. It is an essential for cell division during growth and repair of damaged tissues, while it also ensures that each of new cells receives its own copy of DNA. There are three types of hypotheses for replication process. This process is also called Michelson-Stahl experiment. In this experiment, what happens is in the conservative model, the entire original DNA double helix serve as a template for new D double helix such that each round of cell division produces one daughter cell with a completely new DNA double helix and another daughter cell with completely intact old DNA double helix. One is new DNA daughter helix and one is completely intact with the old one. On the other hand, in dispersal model, it is intermixing has happened. Like, see, the fragmentation of the parent DNA stands intermixing with the newly synthesized DNA. See, this is the parental DNA, this pink one, and the blue one is newly synthesized DNA. Intermixing has happened. So both the hypotheses are not accepted by scientists. In the third model, semi-conservative model, the replication process of the parent DNA double-strand helix results in hybrid DNA. See this hybrid DNA, one newly synthesized strand and one parental strand. Here, each of daughter DNA double helix retains a parent strand and a newly synthesized strand. This experiment of Michelson they used isotopically labeled DNA for this experiment, and it was acceptable. As I already told you, these three models. First, conservative replication. What happens in this replication? Out of two helix formed, one helix contains entirely old or parental DNA, while the other helix contains entirely new DNA. This model of replication is not found to be biologically significant. Scientists believe that parental DNA does not split open up in this model at all. In the semi-conservative replication, it is a biologically significant model of DNA replication proposed by Watson and Crick in 1953. According to Watson and Crick, during the semi-conservative replication, one old DNA strand serves as a template to form the new strand. Hence, each double helix produces contain one old strand every time. Moreover, DNA polymerase enzyme needs a template strand to form a new strand, and there is a possibility of combining new strand with the template strand during replication. So it was acceptable. On the other hand, in dispersive replication, it is a third possible model of DNA replication. In this model, the intermixing has happened. Intermixing of it is a mixture of old parental DNA strand and newly synthesized strand. It was not acceptable. DNA replication will start with the breaking of hydrogen bonds between the two strands by helicase enzyme. See this picture. The helicase enzyme is breaking the hydrogen bonds between the two strands. After breaking these hydrogen bonds, what happens is the RNA primer will initiate the replication process. See, it is initiating the replication process. After initiating the replication process by RNA primer, DNA polymerase 3 will add the nucleotides continuously. See, this is a replication fork. After unwinding the DNA double strand, it is formed like a fork. See, it is called the leading strand and this is called lagging strand. The leading strand is replicated continuously same direction as a moving fork, while the other strand is replicated in opposite direction in the form of short Okazaki fragments. And these fragments see, it is continuous in process and it is in fragments, and these fragments they are also called nicks. This nicks is sealed by DNA ligase. See, DNA ligase is sealing these nicks. At the origin of replication in DNA double strand, helicase will start to unwind the DNA double strand by breaking the hydrogen bonds between these two strands. See, after making after breaking the hydrogen bonds between the two strands, it making a Y-shape like structure. This DNA helicase using the ATP energy and breaking these hydrogen bonds and it makes a Y-shape like structure. It forms two strands, one leading and one lagging strand. Leading strand means it replicates continuously in the same direction like a moving fork. And on the other hand, lagging strand is replicated in opposite direction in the form of short Okazaki fragments. In the leading strand, it forms a continuous in a continuous process. It forms a continuous strand like this. On the other hand, in lagging strand, it forms a short fragments. There will be a space between these short fragments, and these fragments will attach by DNA ligase enzyme. See this picture. After breaking the hydrogen bonds by helicase enzyme, see, this is the leading strand. It replicated continuously like a single strand, and on the other hand, lagging strand, it makes small fragments, and these fragments are called Okazaki fragments. These fragments will be joined by DNA ligase enzyme. These are the steps of replication. In the first step, DNA replication is to unzip the double helix structure of DNA molecules. This is carried out by an enzyme called helicase. It breaks the hydrogen bonds holding the complementary bases of DNA together, A, T, and GC. It breaks the hydrogen bonds between these AT and GC. The separation of two single strands of DNA creates a Y-shape like say a replication fork. The two separated strands will act as a templates for making the new strand of DNA. One of the strand is oriented in 3 to 5 prime direction. This is called leading strand. The other strand is oriented in 5 to 3 prime direction in the opposite direction. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. Leading strand synthesis once initiated occur in a highly possessive and continuous manner. It forms in a continuous way. There will be no stops like a lagging strand. Unlike lagging strands synthesize like in a discrete short DNA fragments. It is also known as Okazaki fragments, which are later joined to form continuous duplex DNA. Synthesis of an Okazaki fragments begin with a primer RNA. It's at the initiation step, RNA primer will initiate the DNA replication, and the DNA polymerase 3 will add the nucleotides to continue the replication.